At SENS Research Foundation, our focus is in advancing therapies for age-related disorders. My name is Amutha Boominathan, and I lead the MitoSense project at SENS Research Foundation.
Every cell in our bodies contains DNA, which is the genetic basis of life. And when we're talking about DNA, what we're usually referring to is nuclear DNA, or the 23 chromosomes that we inherit from our parents. But we also inherit a small amount of DNA from our mothers in the form of mitochondrial DNA, which are small circular DNA molecules residing within the mitochondria in themselves.
Mitochondria perform the important function of energy generation for the cells, and majority of the proteins for this process are encoded by the nuclear DNA and are imported to the organelle.
However, there are 13 very important proteins which are still encoded by mitochondrial DNA and synthesized within the organelle. And several factors make this genetic material especially sensitive to mutations, which cause a number of age-related pathologies. And unfortunately, available treatments for these conditions are currently geared towards symptom management and seldom address the root cause of the problem, which would be actually correcting these mutations. At SENS Research Foundation, the goal of the MitoSense project is to develop an alternative gene therapy-based approach for correcting mitochondrial dysfunction by moving these 13 mitochondrial genes to the protective environment of the nucleus and making them functional.
We have been successful in re-engineering 2 of these 13 genes, namely ATP8 and ATP6, and we recently demonstrated that we can rescue a patient cell line with a particular mutation in the ATP8 gene with regain of function. I would like to thank Lifespan.io for their instrumental role in funding this project.
Now we ask for your support once again as we take these studies to the next level, proving that our technology works in animal models, which moves us one step closer to extending these therapies to humans.
We would like to demonstrate that our engineered constructs can rescue a mouse model called the FVB mouse with a specific mutation in the ATP8 gene.
We have access to this mouse model, and our preliminary studies in the FVB mouse indicate that our strategy does in fact work to rescue complex 5 dysfunction. The money that you donate will be used to perform important preclinical research and will provide partial funding for the creation of a transgenic mouse expressing the ATP8 protein from a safe harbor locus in the nucleus.
We ask that you join us in bringing mitochondrial gene therapy one step closer to the clinic and bringing us one step closer to curing or slowing down age-related diseases. Thank you.